So my name is Mair. I'm a senior fisheries officer and head of the team in the fisheries and aquaculture division of FAO, and I head the team on aquaculture production and innovation. But my specialization is in genetic resources and biodiversity management in aquaculture, and we work together on the genetic resources work. I was asked to start the session today by giving an introduction to a specific tool that FAO has developed called AquaGRIS. And the reason that I'm doing this is that we are looking and working with the Artemia Consortium to actually use AquaGRIS as a tool to categorize and document Artemia genetic resources globally. And that work has started, and I think Liang will talk about the initial work that's been done in China, and maybe Gonzalo will also make reference to it. So we thought it would be helpful if I can introduce what AquaGRIS is, why we have developed it, and how it can be relevant to groups like the consortium. So the outline of my presentation today is I'll provide some basic context as to why FAO has developed this tool. I'll identify what the tool is. It's called AquaGRIS, which stands for Aquatic Genetic Resources Information System. And then I'll go through what it does. So how is it structured, how and who is entering data, and what types of information is it collecting. Then I'll talk about who it's for and why it's useful. We hope it's useful because we've spent the past 5 years developing it. And then I'll finish off by discussing the relevance to Artemia genetic resources, which will be a good segue to the next two presentations in this session. So the context is, and I'm sure you are here at FAO, I'm sure most of you are very familiar with what FAO does, but it's the UN agency that is responsible for leading international efforts on defeating hunger, which is a very lofty goal. Fortunately, we are working on very specific issues. So within the Fisheries and Aquaculture Division, we are looking at transforming aquatic systems to promote the responsible and sustainable management of aquatic food systems. Within the aquaculture thematic area, and you heard yesterday from our Deputy Director, Xinhua Yuan, about the work we do within the aquaculture thematic area, and specifically my role is to lead the team on production and technology issues. But also in the case of genetic resources, we work very closely with the Commission on Genetic Resources for Food and Agriculture, which is another FAO body based in another division, but essentially NFI and our team is responsible for the work on aquatic genetic resources, but the Commission also deals with terrestrial agricultural resources, forests, crops, livestock, etc. So we're part of a bigger program. So essentially we're working to support the members of the Committee on Fisheries and also the members of the Commission on Genetic Resources for Food and Agriculture. Aquatic biodiversity, I don't think I need to preach how important aquatic biodiversity is, but in the specific context of aquaculture, this biodiversity is critical and it's essentially responsible for sustaining fisheries and the growing field of aquaculture to ensure the future supply of food. So if we don't manage our biodiversity, we compromise our future supply of food. Biodiversity, as we all know, is genetic diversity, species diversity, and ecosystem biodiversity. But our work on genetic resources focuses on the first element there of genetic diversity. Obviously, we also deal with species diversity and ecosystem biodiversity. So genetic diversity enables organisms to adapt and change in the wild and to challenges such as climate change, and also to the challenges of adapting to the aquaculture environment and to our changing world. But it's also, in the context of aquaculture, genetic diversity is the building block and the foundation for work on genetic improvement, which in aquaculture is really just starting, and I think the same applies for Artemia. There's a little bit of genetic improvement starting, but we are far, far behind the terrestrial agricultural sectors. The focus of specifically of our work at FAO is on farmed species and their wild relatives, and we call these aquatic genetic resources for food and agriculture, and we use the acronym AQGR, Aquatic Genetic Resources. So essentially that means that, that we have a list of about 700 species, aquatic species, that our member countries have told us they are currently farming. So essentially AquaGRIS will collect information on any and all of those 700 species. I think we currently have, because it's right at the beginning, we currently have data on about 160 species in AquaGRIS, and if anybody's interested to see more of AquaGRIS, we can also do a demonstration at some later stage. But in addition to looking at the genetic resources that are actually used in aquaculture, so the resources that are farmed, we also look at the same species where they occur in the wild, and this is particularly important for Artemia because much of Artemia biomass and cysts is actually harvested from the wild, so it's effectively a fishery. But it is an aquaculture genetic resource, and so we look at characterizing and documenting that resource both within aquaculture and also within the wild, hence the salt lakes, which is the focus of this seminar. So just to give you a little bit of context, we do an awful lot over the past 5 or 6 years, but essentially we've been building a program. We started off with some guidance for governments on the essential criteria of what resources and infrastructure is needed to manage genetic resources. Then a key achievement which was finished just after I arrived at FAO is that we produced the first report on the state of the world's aquatic genetic resources. That's called a global assessment. Then we looked at some of the needs and challenges that were identified in that report. It's a 260-page report, and we have hard copies if anybody's interested in it. But one of the issues that was raised was about information. So at that time, in 2019, we had already started to think about an information system on genetic resources. It didn't become named AquaGRIS until last year, but that's essentially what it was. We have lots of other resources, including e-learning programs on genetic improvement, case studies on the importance of genetics, etc. But in terms of the cycle of work we do with the Commission, we translated the needs and challenges identified in that global assessment to a global plan of action, or a GPA. That global plan of action was published in 2022, and it has been adopted by all FAO member countries by the FAO Council. And so essentially it's an instrument, it's a non-binding voluntary instrument that all countries have signed up. And it's now a tool that can be implemented. I won't go into the details of it, but essentially it has 4 priority areas. Inventory, characterization and monitoring is the one that AquaGRIS is designed to support countries to deliver. But we also have a priority area on conservation and sustainable use, which is probably the area most relevant to Artemia. Then we have a priority area on development, which will become, so that's genetic improvement, that will become increasingly relevant for many species, including Artemia. And then we have the 4th one on policies and institutes, which have to underpin all the other 3. So having done all of this work, we are now in a phase where we are trying to support countries and organizations such as NGOs and intergovernmental organizations to start implementing so that we can actually look at changing and improving the way in which genetic resources are managed by countries. So we're now in the exciting phase that we are trying to work with countries to support the implementation of elements of the Global Plan of Action. But in relation to AquaGRIS, which is the focus today, we produced a prototype which was released in 2022. That one looked only at farm type, so only genetic resources used in aquaculture. But following feedback on that and some piloting, we redesigned and improved it, and we have just released the full version of AquaGRIS in June 2024. And in fact, we will be launching AquaGRIS in an event on the 17th of September, just before our next Intergovernmental Technical Working Group. So it's that full version that I want to talk to you about today, and it's basically ready, it's used, it's got some data in it, but we have a huge journey to work with countries now and organizations and experts to populate it with genetic resources, information on genetic resources. Just to put that all in context for you, the framework, AquaGRIS, the Global Plan of Action, our training resources, They can be all used to support countries to essentially create things like a national registry of genetic resources, key conservation needs, priority farm types for development, genetic improvement needs, capacity building, etc. And ultimately, collectively, they can be used to support national strategies to enhance the genetic management of genetic resources. But I'm talking today about AquaGRIS, and the principal function of AquaGRIS is to help countries to build national registries of aquatic genetic resources. So why do we need an information system for aquatic genetic resources? I've talked already about the fact that the State of the World report, the global assessment in 2019, identified the need to establish and strengthen national and global characterization, monitoring and information systems for aquatic biodiversity use in aquaculture. It identified that there is a lack of a characterization and specifically monitoring of the status of aquatic genetic resources, and that is particularly so at the level below the species. Countries report to us about the species that are, or species groups that are being farmed, but they don't, most countries don't even collect any information about what genetic resources, what strains or varieties are being used in production. So there's almost no information at that level. There are a few national systems, Germany, China, India. They have systems, but these are not harmonized. Each system is designed for a different purpose. It has a different scope, and it has a different way of describing genetic resources. So essentially, there's a role for a harmonized system, and it's a very good role for FAO to develop that. But even within FAO, our systems are not consistent. So some of you may have heard about FishStatJ, which is our information system that provides data on the production of aquatic species. But in some cases, they group species together, or they have groups of species that are not specifically identified, and they even do have a few genetic resources below the level of the species, such as hybrids, but they are not correctly categorized. And additionally, terminology is absolutely not standardized. So we're probably all aware of the names of genetic resources. So a species I work with a lot is tilapia. You can go to many countries in Southeast Asia and people will tell you about a tilapia that has a name. Sometimes that name is directly related to its status as a genetic resource, but very often it's nothing to do with the genetic status of it. And yet people consider it to be a strain or a breed. And then we also have these different terms being used: landrace, line, breed, strain, etc. They're non-standardized, and we found that when we did the global assessment, that the way people describe genetic resources was different. So one of our goals with AquaGRIS is to standardize the way in which we describe and name genetic resources. It's important to recognize also that FAO plays a role with similar information systems in other terrestrial genetic resources. They have been having a system for crop genetic resources and livestock genetic resources for over a decade now, and there's also one being developed for forest genetic resources. So again, in the aquatic sector, we are playing catch up. So this is what AquaGRIS looks like. I'll go into a little bit more detail, but essentially there are two user interfaces. We have a data entry user interface and a dissemination user interface. And I see people taking pictures of the slides. We're quite happy to make the presentation available through the organization. So I put a little bit of detail in there that I won't be describing. But essentially, the data entry user interface is where experts who have the knowledge about genetic resources will enter the information. Because we are FAO and we have to work with official government representatives, we have in many countries a national focal point. And that national focal point is our key contact for entering information about any country. But we recognize that no individual in any country knows everything about the genetic resources. So we rely on those national focal points to designate experts. In most cases, those experts will be national experts, but in some cases, and Artemia may be that example, that it can be a mix of international experts and national experts to enter the data. But essentially, that's not open to everybody. It's only open to people that are registered to enter and are approved by national focal points and or by FAO. Once the data is collected there, and I'll talk a little bit about the kind of data we collect, that enters then a data registry. And at that point, the data in the long term should be validated by the national focal points, because once it's validated, it's published and it's in the public domain, and we should be making sure that each national focal point is happy that the data is accurate. Currently, because we are only starting to enter data, we are not doing the validation yet. So the current data in AquaGRIS should be viewed with a disclaimer that it's not yet validated by national focal points. But then the interface that's available to the public, anybody in this room can access it, and the link is there in the bottom corner of the slide, is the dissemination user interface. And this is where users can enter the system and find out all of the information that's been entered into the system. You can do that through specific search functions such as species fact sheets. So you can generate a fact sheet on Artemia franciscana, for example, and that can give you global information available in the system on that species, or you can filter it down to a particular country or a region. Similarly, we have country fact sheets, so any country can produce a fact sheet on all of its genetic resources or just some. We also have the capacity to generate customized reports, which essentially you can drill down into details on every species and every farm type within those species. And the next tool that will be added to the system is the capacity to generate indicators. So the idea of having indicators is that for the first time ever, we'll be able to monitor the status of our genetic resources over time. I have a brief slide that I show you at the end about what those indicators are covering, but some of those indicators will definitely be useful for Artemia genetic resources. And those indicators apply to those 4 priority areas of the Global Plan of Action that I outlined earlier on. So we enter data on a national basis. So we're looking at a genetic resource only under a national jurisdiction. So we could have the same genetic resource in 2 countries that will be entered, or same or similar, that will be entered separately by each country. We deal with all of the major taxonomic groups, finfish, both marine and freshwater, mollusks, crustaceans, macro and micro algae, and obviously zooplankton as well, and even miscellaneous species like crocodiles and frogs. As I mentioned earlier, we cover around 700 species. If the species isn't in the system, you can request FAO to add it if you can demonstrate that it is a cultured species. So it must be in use in either commercial or subsistence aquaculture. If it's just at the level of research for aquaculture, we don't include it yet. And then we deal with both farm types and genetic stocks, and these are very important terms that I'm going to define for you in a minute. So with the data entry interface, we start at the species level, so you start entering by species. We have generic level information, and then we have aquaculture related information and wild stock related information. Under the aquaculture related information, the key expertise we need is to be able to determine what genetic resources and how they fit the criteria for describing genetic resources, which we allocate as farmed types. And we have primary farm types which relate to the status of domestication and development. And then we have secondary farm types, which is added value technologies such as hybridization, triploidy, monosex, and things like that. And then for our wild stock information, there are 2 key. The most important thing is classification as a genetic stock, but we also look at the management units and how those genetic stocks are managed. And so there's another critical expert input required here to identify which are the genetic stocks that meet the definition of a genetic stock, which I will provide in a minute. So the species level information that AquaGRIS collects is the name and the local name, whether it is native or introduced. We have a question about whether it is being impacted by climate change, and that basically requires there to be some evidence that there is an impact of climate change. And then with the aquaculture related information, we collect production information. And Artemia, when we did the field testing of the first version of AquaGRIS, which Patrick supported, was quite interesting because it became evident already that there's 2 products from Artemia. There's biomass and there's cysts. So as a result of that, we actually changed the system to allow the entry of 2 or more products. So if you're entering information on Artemia, in some cases the primary product may be a biomass product, and in another case it may be cysts. and so we can cover both of those. We look at production trends, and then we look at the genetic resources below the level of the species, which we are determining farm types. In the case of wild stock information, if the stock is fished, then we have production information from the harvesting, which is obviously the case in Artemia. Maybe we don't think of it as fishing, but it's the same thing. It's harvesting from the wild. We look at the status of the habitat range and whether it's contracting and what trends there may be in the habitat range. And obviously the concern here is that one of its principal habitats is contracting. And so that information would be recorded. We look at extinction risk. We look at the IUCN categories, local red lists, etc. Then we look at the management assessment units, but most importantly, we characterize the genetic stocks occurring in the wild. So looking at the farm type information, a farmed type is a descriptor that is applied to farmed aquatic organisms at the level below a species. So it includes farm types such as strain, which we apply to animals and microorganisms, varieties, which applies to algae and plants, and then secondary farm types like hybrids, triploids, monosex, even genetically modified, gene-edited, for example. And so every genetic resource that you find in a farm should be able to be described at two levels. Firstly, what species is it? And secondly, what farm type is it? So everything you find on a farm should be allocated to a species and a farm type once it's been defined in here. So I want to talk about primary farm types. I won't go into secondary, it's not so relevant for Artemia. A primary farm type essentially relates to the level of domestication of the animal. So if it's used in aquaculture, but it's essentially harvested from the wild, then we call it a wild-sourced farm type, and that's where it's essentially either the broodstock or the seed itself is captured in the wild. So in the case of tuna aquaculture, we very often harvest in the wild and we fatten them up, so that's a wild-sourced. At the other end of the spectrum, we have strains or varieties, strains for animals and microorganisms, varieties for plants and algae, which have been changed in some way, either by long-term domestication or by selective breeding, and they've been changed to the point that they are now differentiated from other farm types of the same species within your country. So the classic example would be for maybe the Norwegian salmon, which has been genetically improved for 10, 15 generations. It has distinct characteristics, and each one has its own distinct characteristics. So we estimate somewhere between 10 and 15 of global aquaculture production is made up of these developed farm types. Then what do we do with everything else in the middle that's been domesticated? It's no longer in the wild, but it's not actually developed to the point that it can be differentiated from anything else, either by its performance or by its phenotype. So we coined the new phrase captive propagated. So essentially that covers everything in between the developed farm types and the wild sourced. And that's probably the majority of the farm types that are being currently entered into AquaGRIS. This is a definition of the primary farm types. I'm not going to read them out, and I'm happy I will distribute this. The definitions are available online. It'll take it too long. But each one has a strict definition, and so when you're entering information into AquaGRIS, you look at this definition and you try to identify which genetic resources fit which type. Looking at the wild genetic resources, the key definition here is the genetic stock, and this is the one most relevant, I think, to Artemia. And so I will read out this definition. It is a stock having one or more distinguishing and heritable characteristics that distinguish the wild stock from other wild stocks of the same species within the same country. These characteristics could be genetic differences, but that usually requires some research to have been carried out to actually do a genetic analysis of those, looking at molecular signatures. But it can also include distinct morphological and phenotypic characteristics, such as cyst size, growth rates, etc. And then it could also have proxies for genetic differences, such as geographic and reproductive isolation. So if you have a salt lake, and you know that the Artemia in that salt lake are unlikely to have had any gene flow with any other population of Artemia, then even if you've done no genetic analysis, you can make an assumption that based on what we would believe about evolution, that it is almost certainly a genetic stock. So you don't have to have the genetic data, but it's obviously ideal because that can then confirm it. And then the management or assessment unit is essentially who, if anybody, is managing that genetic resource. So that would include things like a fisheries management system, a conservation unit, an identification of evolutionary significant unit, whatever. And it's an interesting discussion for us about how genetic resources in salt lakes are actually managed. And we can maybe talk about that in our discussion. So I mentioned earlier on that we have these indicators. I won't go through them. We actually have now, and we are going to be presenting for adoption by members of the Commission, a series of indicators. All of these relate to the genetic status of the resources, and they can definitely be applied to Artemia, either on a national basis or a global basis. So I imagine, for example, that the Consortium, once we are able to enter information on Artemia genetic resources, that can be monitored, and these indicators for Artemia globally, regionally, can be generated every few years, and you can see what progress is being made. But the indicators are designed specifically for looking at progress against the global plan of action, and they can be applied by countries to look at that. But they could, or some of the indicators could also be applicable to, for example, the Kunming-Montreal Global Biodiversity Framework. One of their headline indicators is effective population size. We are trying to collect that information. It's not easy for many species. So who is AquaGRIS designed for, and what can it be used for? Well, we consider that it's a range of stakeholders that it's relevant to. Principally, it's designed for resource managers. They can most commonly be national resource managers, but also to policymakers, to aquaculture producers, to researchers, and international organizations such as the Consortium. And AquaGRIS can support stakeholders in a range of different purposes. It can be used for standard categorization of aquatic genetic resources, such as might be included within a gene bank, which I think is relevant to Artemia. It can be used for developing national strategies, for example, to implement some elements of the global plan of action. If you understand the resources you have, you can work out where to prioritize your effort in managing genetic resources. It can be used for preparing national status reports, which FAO can then aggregate into global status reports. It can be used for making data on farm types available to producers. If I'm a farmer in a particular area for a particular species, I can go into AquaGRIS and I can look at the characteristics of the resources that are available to me in my country. It can be used for developing registers of aquaculture producers. It can be used to develop R&D plans and roadmaps. It can be used to support research and publication of research, and it can provide scientific advice to policymakers. So it's quite a broad church. We believe it has applications to many different stakeholders. We actually also believe that it's actually more diverse and broader than the information systems developed for crops and livestock, and we have the opportunity to do that because we have so many fewer genetic resources in aquaculture than we have in terrestrial agriculture. So on to the most relevant bit of Artemia. How can this be relevant to genetic resources and Artemia genetic resources? I hope by now that you've already seen possible applications in whatever is your role as a stakeholder in Artemia. But first of all, it represents a standardized system for characterization of both native and non-native genetic resources, and we will hear a little bit later about how AquaGRIS is being used to characterize Artemia resources in China. This can be done at the level of farm types in aquaculture, but also the genetic stocks. It's probably the genetic stocks that are most relevant to this symposium and to the salt lakes. It can be used to create global, regional, and national registries. It can be used to link gene bank records, regional, global gene banks, back to national resources. So there's a common link. Everybody understands when you use a particular name of a farm type or a wild or a genetic stock, that it's in the system and everybody's talking about the same thing. It can collate aquaculture information such as production of biomass and cysts by species. It can be used to monitor production trends in aquaculture, and it can be used to provide information on characterized farm types. On wild stock information, we can look at the production levels of harvesting of biomass and cysts. We can look at the trends in production. We can look at threat levels, particularly in relation to extinction risk. We can look at how stocks are being managed, and then we can actually look at the individual characteristics of genetic stocks. So looking at the information that you can find we enter and you can find in AquaGRIS on farm types and genetic stocks, information that applies to both of those are information on production. Typically, we don't look at absolute production because quite often that is not monitored, but we look at production as a percentage of national production of the entire species. So we can have an idea of whether a farm type is making up 5% of national production or 50% of national production. We can find out about the origin, whether it was introduced and where it was introduced from and when. We can look at the risk of a farm type or a genetic stock compared to others. So if we do a genetic improvement and we produce a new farm type, is the risk associated with that farm type and its impact on biodiversity, for example, is it greater or lesser than the risk of the species as a whole? Then, probably most importantly, we have information on the characteristics of the genetic stock or the farm type. And in the case of Artemia, this can be cyst size, nuclei size, nutrient composition such as hoofer content, and I see already that we have data on that in AquaGRIS. We can look at the conservation and whether genetic resource is held within a gene bank. We can effective population size if it's been estimated, and we can look at germplasm exchanges. So if there's a genetic resource, AquaGRIS can record if that genetic resource has been transferred to other countries. For data that's specific to farm types, we can look at the number of generations under domestication or selection. And for strains, we can look at how they were developed, what kind of selection has been used, documented breeding history, breeding program implementation, etc. And in specific information for genetic stocks would relate to the management and assessment of those stocks and the status of the stocks, so whether they're in decline, stable, etc. So that's a summary of the information. These two links, and as I said, we're happy to make the presentation available. The first one is information about AquaGRIS on our website, and the second one is actually the access to the AquaGRIS dissemination user interface. So the second link there is the one you can go into to have a look at the information that's already there on AquaGRIS. I should caution that we're really at the beginning of this process. Over the next 5 to 10 years, we want to work with countries, as many countries as possible, to start populating this. And the more data we get into AquaGRIS, the more powerful it's going to become. And I think if we do persuade countries, and countries asked us to produce this, if we do persuade them to enter data and creating national registries, if we can get 50 or 100 countries with national registries, I believe AquaGRIS will become an incredibly powerful information resource. But I think even for specific species groups, and what we've been talking about so far is how can we work with the consortium to upload this information on Artemia. So even if countries don't have full national registries, if we are supporting countries to put in information on Artemia, we can have a full global picture of Artemia even without a full national registries of all the countries that have it. So I think it can be applicable outside of just the national registry. So I'd like to thank you very much for your attention. As chair, I can invite any questions, but maybe we can also, in a broader context, after the next two presentations, we can have a discussion, because I think both of them will probably touch on the use of AquaGRIS. But do you have any specific questions for clarification about AquaGRIS and how it works and what I presented, then I'm happy to answer.